Hello, I'm Tom Gardner from the University of Michigan and the Mary Tower Moore Vision Initiative. Here in session 3, we're going to discuss assessment of function of the retinal neurovascular unit. And we have 3 outstanding contributors. The first is Dr. José-Alain Sahel from the University of Pittsburgh. The second is Dr. Jason McAnany from the University of Illinois in Chicago. And the third is Dr. Lesmes from AST in California. Dr. Sahel will be discussing measuring functional vision through mobility testing and virtual reality and the learnings from his studies in inherited retinal diseases.
Thank you, Dr. Gardner, for inviting me to present on what we try to develop in terms of outcome measures in retinal degeneration. So my talk is not going to be focusing on diabetic retinopathy. I've been working on retinal degeneration for many years. These are my disclosures. Some are relevant to my talk because of the gene therapies we were developing. I've also been working with a lot of people that I cannot thank now, but some of the names are on the slide and some of the pictures on many of these projects. In retinal degeneration, we are facing challenges that probably are relevant for diabetic retinopathy also. We have to tackle the cause of the disease, and in that case, we are trying to tackle that using gene therapy to correct the gene defect. We are at some point trying to protect the remaining cells in the retina, and this is neuroprotection, which is probably a very important topic in this field. And there are stages of a disease where the vision is so low that even protection is no longer relevant. The target cells are dying or are dead. And this is where you want to restore vision using either prosthetic vision or optogenetics or stem cells. But the challenges are multiple. Some are safety challenges. Some are relating to how advanced the stage of the disease is. Some are relevant to whether we want to be palliative or curative. Some are really, are we tackling the cause of the disease or an amplifying pathway? And you can really list the question, but the key questions are really, how do we demonstrate activity? How do we demonstrate that we are restoring vision? And what do we call vision? Is this the type of visual function we test in the clinical setting? Or is this the functional vision that people are using in daily life? And there was a big success a few years ago with the gene therapy in retinal degeneration, thanks to the work of many people, especially Jean Bennett and her group, were leading to the approval of the first ever gene therapy called Luxturna. And this was based mostly on the functional vision testing or mobility testing. I'm going to show you a movie of the first patient we treated in my group in Paris, which was the first also in Europe, a young girl affected with this RP65 mutations. And she's trying to move in a low luminance level environment, a platform I'm going to talk about in a moment called Street Lab. And she is very slow. She misses many parts, takes her a lot of time to navigate because she has a very limited vision. She normally should follow the dots on the floor. She doesn't see the dots. And she's being guided very slowly through all that. And this is before the therapy. And this was her life before she got the gene therapy. And you have seen many of these movies, but what you will see now is what happened after she got the therapy, a few weeks after, and she's asked to do exactly the same task. And you will see the difference in a short minute, less than that, hopefully. As you will see, she's now able to navigate on her own very fast and performing. And this video, actually, I have 20 videos of that. Many patients have the same type of outcomes. But we were hoping when this happened that this would be really the start of many therapies. And actually, this is the only therapy that has been approved so far because there is a sort of valley of death, many failures in gene therapies, many difficulties to get approval from the agencies, raising a lot of questions on what is the preclinical package, what are the outcome measures, what are the expectations, how can we design trials that are meaningful, and how do the agencies react to the questions we have and understand the challenges we are facing. And this leads to the fact that there are currently very few gene therapy trials. And as I said, currently not yet any other gene therapy approved. How do we face that? There are many ways to face that. And I just want to propose on one side, more science, more understanding of the disease and what we are expecting from the therapy. On the other side, an improved dialogue with the agencies. For example, the CBER, the previous director, Peter Marks, offered a platform approach, really trying to capitalize upon the knowledge from one disease to the other. But this is not enough. We need to build a body of knowledge, for example, on natural history. And the example was given by Foundation Fighting Blindness, clearly by the DRC on net many, many years ago. And this is modeled after it, actually, trying to understand the evolution of a disease in multiple patients across the world. And this led to analyzing several gene defects. And now we have a universal study trying to detect what is the magnitude of change that is relevant versus the variability of change, the signal to noise, what is a real change. And this is really the repeatability that we are observing, the sensitivity to change in the population and trying to eliminate the flooring effect, but also the ceiling effect, and what is meaningful for patients. So I will not spend too much time on that. And I will just want to acknowledge many people that were involved in that. My group has been focusing on developing what we call performance-based testing, which is demonstrating the ability to help the patient in daily life. And we tried to use multiple scales, and I just lead the type of testing we use in clinical trials usually, and trying to focus on the type of test that we develop. One of these is a virtual reality testing that we use to analyze the mobility. You saw the maze for the mobility testing that was used by Spark. And initially, this was based on the Street Lab, the platform in Paris, where we are monitoring the impact of low vision in mobility of our patient. This is an example of a patient with low vision, of retinitis pigmentosa, the trajectory of a patient, the direction of a gaze, the ability to move around that, and the ability to measure all the type of movement, the gait, the gaze, the trajectory, the head movement, and demonstrating actually a very reproducible responses. For example, the amplitude of the saccade, the rotation of the eyes. We use that to build the most mobility testing, which is virtual reality testing, which was compared to a real-life mobility testing. And this has been achieved in our group in Paris and now replicated also in Pittsburgh through 3 steps. 1 step on healthy volunteers, 1 step on patients that were the first testers, and then the validation that occurred over time to really validate at multiple levels, multiple experimental sessions, showing that 20 mazes were needed to avoid any learning effect, and 5 sessions of learning were good enough. And this is a demonstration of how consistent it is across multiple sessions, how it does reproduce in brown and in green what is in real life versus the maze in virtual reality, how it does correlate to multiple levels of testing, real life and virtual reality, how it does correlate with different type of tests like visual field, visual acuity, and these are different studies that were conducted, comparing, and this slide is very busy, but trying to analyze cone function using different methodology like the octopus, the Goldman, the cone rod breakout in dark adaptation with very good correlation. And based on that, we were able to develop this test, which is now showing a very good level of reproducibility that has been adopted for the natural history studies for Foundation Fighting Blindness, but also by several companies for clinical trials, and hoping it will be an approved endpoint. It's exported, it's got accepted, and patients say that it is reproducing what they are feeling in daily life. We also developed other tests, for example, central vision testing, object recognition, object detection. This is called VISA. People have to detect a cup or an object. Here, this is a very small, round, red object that we have to detect. And this has been also a very consistent test, which is now being standardized with the ability for patients to explore the room. And because we're leading with very low vision patients, we developed a test for extremely low vision mobility. That is a physical test, but now also a virtual test that is reproducing what has been observed in daily life. So this is the work of many people at the team in Street Lab, especially the work of Cola Autier, but many others who I want to acknowledge. And we are now reproducing that and exporting that. And we believe that this type of testing, correlated with patient-reported outcome and the functional vision testing, has a future in any type of retinal and maybe ocular disease. Thank you very much. And now I'm very pleased to hand back to Dr. Gardner.
Thank you very much, Dr. Sahel. And now we have Dr. Jason McAnany, who is going to talk about mechanistic insights from visual function testing in diabetic retinal disease.
Well, thank you, Dr. Gardner, for the introduction. So today I'm going to tell you about mechanistic insights that we've gained from visual function testing in diabetic retinal disease, or DRD. So as a bit of background, there's now mounting evidence of neural dysfunction that can precede the clinically apparent vascular changes in DRD. And it's now thought that diabetes impairs the neurovascular unit. In the clinic, we have several approaches that are available to assess vascular abnormalities, but approaches to quantify neural abnormalities are not as well established. So this has been a major interest of our group, and we've been very interested in using non-invasive methods to assess visual function in DRD. And the approaches that we've used can be loosely grouped into three different categories. So we have electrophysiological measures. These include the full field ERG, which I'll tell you more about in a moment, the multifocal ERG, and the visually evoked potential, or VEP. The ERG measures target the retina, so we can localize any disease or abnormality to the retina itself. The visually evoked potential requires both retinal processing and cortical processing. We've also been interested in pupillometric measures. The pupil measures require both retinal processing and also subcortical processing. And finally, we have psychophysical measures. These measures assess function throughout the entire visual pathway, from the photoreceptors all the way through cortex, and they require a behavioral response. So these include visual acuity, contrast sensitivity, visual field perimetry, color vision, dark adaptation, among others. So you may be familiar with these types of measures. So given the limited amount of time I have today, I'm going to focus on two measures, just to give you an example of how we can use non-invasive techniques to better understand visual function in DRD. So I'll tell you a little bit about the full field ERG and also about contrast sensitivity. So that we're all on the same page, I'll first describe what an electroretinogram, or ERG, is. So the ERG is an electrical response that's generated by the retina in response to flashing or flickering light. So to record this response, we use a fine fiber electrode that lays along the lower eyelid in contact with the cornea. The patient looks into a large dome that looks like this below, and the patient then can see a flashing or flickering light that looks something like this. The response of the ERG looks like this waveform. So what I'm showing you here is the voltage, or the change in amplitude over time that's elicited by the retina in response to this flickering light. So what I'm showing you here is a response from a large group of visually normal, healthy control subjects. And you can see that every time there's a flash of light, both a trough and a peak are generated. If we have 62 flashes of light in a second, then we'll have 62 responses in a second. So you can compare this healthy, normal response to that of two different diabetic groups. So I'm showing you in green a group of diabetics who have no clinically apparent retinopathy, and a second group of diabetics who have mild non-proliferative diabetic retinopathy. And you can see that the amplitude, or the size of this response, is about half of the control group. And again, we can measure this now from the trough to the peak to give us an amplitude. In this slide, I'm plotting the amplitude of that ERG, so how large was the response, across many different temporal frequencies, from very slow flicker over here around 10 hertz to very fast flicker over here around 100 hertz. And there are two aspects of this plot that I'd like to point out. The first is that at high flicker frequencies, the control group, shown in black, have a larger response than the two diabetic groups. In fact, the diabetics with no clinically apparent retinopathy have about a 40% reduction in their amplitude, shown here, and that diabetics who have mild NPDR are even more affected. They have about a 60% loss of amplitude. You can compare this with the responses that we found at lower flicker frequencies for slower flicker. So at slower flicker, there are only minimal differences among the three groups. And in fact, they're almost identical for 10, 15 hertz. So what we're finding is that there's a selective loss of high frequency flicker in diabetics. So what can we do with this information? Well, I think this might be useful for better staging ERD. And I'll show you what I mean. So this is our standard way of thinking about diabetic retinopathy. We usually classify these patients according to the retinopathy stage, from no DR, mild, moderate, severe, or proliferative disease. We also consider the extent of diabetic macular edema running this way. So in the data set that I showed you, there were 20 patients who had no clinically apparent retinopathy, clustering here, and 20 patients who had mild NPDR, clustering here. So by and large, these are very similar patients. There are not marked differences among these individuals. Now I can add another axis to this plot. And this axis represents the extent of neural dysfunction that we quantified from the flicker ERG that I showed you a moment ago. So now you can see that despite their very similar clinical characteristics, these patients vary tremendously in terms of their neural dysfunction. Some of them are perfectly normal, falling in this range. Others are 2, 3, maybe even 5 standard deviations below our normal range. So we can use the flicker ERG perhaps to better stage DRD. The one major limitation of the ERG is that it only tells us about retinal function. It doesn't tell us what the patient sees or experiences. And because of this, the FDA has been somewhat reluctant to use the full field ERG as a primary outcome measure in clinical trials. So to address this concern, we've begun measuring psychophysical flicker sensitivity in DRD. So to do this, we developed a novel visual field perimeter, which functions much like a normal Humphrey field perimeter, except that the light flickers on and off at a given rate in the back. So here's an example. So if this light in the back will begin to flicker on and off. Hopefully you can see that. And we can change the intensity of this flickering light. We can make either more visible or less visible, which allows us to measure a threshold. And in fact, that's what I'm showing you here to the right. So this is a contrast threshold for our control group in black, and then for our diabetic group in red. And you can see that when the light is flickering relatively slowly, there are only minimal differences between these two groups. Now, if we flicker the light more rapidly, you see that there's a difference that begins to emerge, where the diabetics have less sensitivity to the flickering light than the normal controls. At our highest flicker frequencies, we find there's about a 28% loss of flicker sensitivity for the diabetic group compared to the controls. And if you look at this curve, you'll see that this looks very similar to what I showed you a moment ago for the ERG, where there are only small differences at low frequencies and larger differences at high frequencies. And in fact, we can correlate these. So we can take our ERG response and we can correlate that with our psychophysical response. So each of these dots represents a different patient. And you can see there's an excellent correlation between the ERG sensitivity and the flicker sensitivity from the psychophysical measures. So what this is telling us is that diabetics often do not perceive flicker as well as visually normal controls. And we think that this abnormality might be arising at the retina itself, perhaps within the cone photoreceptors. So to conclude, this is just one example of how we can use a non-invasive measure to learn more about neural dysfunction in DRD. And the flicker data that I showed you provides some mechanistic insights into neural dysfunction in these individuals. The selective loss at high frequencies we think is consistent with a cone sensitivity loss. An assessment of neural function might provide a better basis for understanding disease mechanisms and for perhaps classifying DRD. So thank you for your attention, and I'll turn it over to Dr. Gardner again.
Thank you very much, Dr. McAnany. And now, Dr. Lesmes will discuss contrast sensitivity testing in DRD.
Thank you, Tom. And thank you to the meeting committee for the opportunity to speak today about contrast sensitivity function in diabetic retinal disease. I'm especially happy for the chance to contribute to this panel led by Tom Gardner and to speak alongside such respected colleagues as José-Alain Sahel and Jason McAnany. Thank you to Jenny Sun for this representation of diabetic retinal disease and how it highlights the need for an updated staging system. If we consider the trajectory of disease in this multidimensional space, from the upper left to the bottom right, we can see the trajectory from the good outcomes that are possible with systemic health to the poor outcomes that are realized with the worsening of neural, vascular, and biochemical function. In a new staging system, it will be critical to recognize how these multidimensional variables interact to contribute to the worsening of visual function and the deep reduction of quality of life that can occur for people with diabetes. I'd like to highlight some parallel efforts by the Ryan Initiative, led by Vas Sada, specifically their efforts to update the staging of age-related macular degeneration. In this report, they conceive of phenotypes of retinal disease as multidimensional data clouds with both non-modifiable and modifiable risk factors. Their proposal is to study the longitudinal flow of these data clouds, to evaluate how they may move over time, and to use that information about disease to improve drug development in AMD. But to return to the Mary Tyler Moore Vision Initiative and the Visual Function Working Group led by Adam Glassman, we can think of their job as zooming in on this dimension of visual functions and trying to figure out the most interesting subspace needed to quantify vision loss across the stages of diabetic retinal disease. In one example, imagine that a focus on this visual function axis is followed by zooming out on variables like the mobility testing presented by José-Alain, or the electrophysiology presented by Jason, or the contrast sensitivity function that I will discuss today. The working group noted that because BCVA is affected only late in the disease, that one primary task was to rank other endpoints with the potential to quantify vision loss across all disease stages. Contrast sensitivity function emerged as one candidate, but over the next 5 years, more evidence generation is needed to make it viable as a registration endpoint for diabetic retinal disease. So let's talk about how we measure contrast sensitivity. This map of visual patterns, defined by the two critical dimensions of size and contrast, is the foundation of our assessment of contrast sensitivity function, which is the two-dimensional boundary that separates patterns that are visible and those that are invisible. On the right of this map, visual patterns are small. On the left, they are large. On the bottom, they are high contrast and are easy to see. And at the top of the map, they are low contrast, difficult, and sometimes impossible to see. One hallmark of this function is that it peaks at intermediate sizes and sensitivity falls off both as the patterns get smaller and as they get bigger. How will we measure this map? And the answer is 3 letters at a time using active machine learning. When 3 letters are presented on the screen, the first is the easiest to see, the last is the hardest to see, and the one in between is in between. When the patient reads the letters, they're marked on the remote as correct or incorrect, and when that information is put back into the active machine learning algorithm, it comes up with 3 new test conditions, which is then followed by another 3 test conditions, and on and on. This threshold contour that is measured can be summarized by a few metrics. One is the area under the log CSF. The other is a low contrast VA. And to review on how these metrics have performed in diabetic retinal disease, we can see that in terms of signal and noise, that we have noise of 1.5 dB and signals of 2.5 and 5 dB for mild and moderate stages of NPDR. Also with importance for noise, we have a low patient burden. This means that with no chin rest and normal seating, testing can be done in 4 minutes. With 95% of the tests that we've done in diabetic retinopathy, which here are numbering near 1,600, 95% of those tests are between 3 and 7 minutes. I want to take another short digression to talk about the prominence of the contrast sensitivity function and how its shape is conserved across many vision and imaging systems. If we start with the results that were first presented by Cynthia Ousley in a Now Classic paper, we can see that the hallmark shape of the function, with a peak in the middle and a fall off on both sides, is conserved in birds, humans, cats, primates with induced amblyopia, across different neural stages, which include retinal ganglion cells, midbrain, and primary visual cortex, in functional brain imaging experiments, and also in deep neural networks that, when trained on an image set and later tested with gratings, show a contrast sensitivity function that is shaped just like in humans. To return to what's needed for clinical trial endpoints, in a previous meeting of the Mary Tyler Moore Vision Initiative, Adam Glassman presented several criteria that are needed for clinical trial endpoints in DRD, such as the characteristics of measurement, correlation with disease, and reliability. I'm happy to report that when it comes to contrast sensitivity, we've shown strong correlations with quality of life and daily activities. We've shown sensitivity to disease stages, such as mild and moderate NPDR and DME. In addition to the work done by Jason McAnany, I want to acknowledge how much of this validation work was done by John Miller and the Retina Service at Mass Pioneer, who have evaluated how contrast sensitivity correlates with patient-reported outcomes, with vascular structure measured with OCTA, and then the normative and test-retest studies that demonstrate a favorable level of noise for the size of the signals we'll be measuring in diabetic retinal disease. In terms of evidence generation over the next 5 years, here is a roster of studies that will be evaluating the longitudinal patterns of contrast sensitivity during disease progression and treatment. We're especially excited for the prospects of the MAGIC study with Charlie Wyckoff and the Retina Consultants of Texas to compare contrast sensitivity with retinal non-perfusion metrics that are serving as the primary endpoint, in addition to protocols AR and AS that were designed to validate functional endpoints for diabetic retinopathy and diabetic macular edema. To summarize, diabetic retinal disease should be evaluated across multiple dimensions. Contrast sensitivity function is an important dimension for spatial vision that shows potential as a visual function endpoint. And lastly, ongoing studies will contribute to the knowledge that is needed to validate endpoints for registration trials. Thank you so much, Tom, for the invitation to speak to the committee and the panel today.
Thank you very much, Dr. Lesmes. And now we will have the panel discussion.